Good afternoon. I'm Dr. Mark Berger, Chief Medical Officer at Genprex, and I'm going to be able to share some of the exciting things that Genprex has been doing in the gene therapy field. We have a brief forward-looking statement, and then I'm able to summarize what we've been doing in gene therapy platform for oncology. We have a separate gene therapy effort in diabetes. Our gene therapy in oncology is important because it's the first systemic gene therapy that's used for the treatment of cancer in humans. We're focusing on large markets and unmet medical needs. Lung cancer is the focus of our gene therapy program in oncology, our initial focus. We're using combination trials with top-selling cancer drugs, as we've been able to demonstrate synergy with those drugs. We have two trials, one open and one about to open shortly, and both of them have FDA Fast Track designation. We've demonstrated clinical achievement in previous trials with our initial drug called Recorsa. As I mentioned, we've initiated one of the trials, and the second will initiate shortly. We have world-class academic partners, both in terms of providing us advice, as well as participating in the clinical trials. Expanding pipeline, as we've been able to expand our initial indications to small cell lung cancer. And in addition, we recognize that there are other tumor suppressor genes that we can utilize. We have a strong balance sheet that can carry these programs forward. And as I mentioned before, we have a preclinical program in diabetes that I'll briefly touch on as well. This is an overview of our research and development pipeline and showing that the two lung cancer studies we're calling a Claim 1 and a Claim 2, which I'll speak about. First, to focus on the oncology program, since it's the more developed program. What we're doing is utilizing tumor suppressor gene, in this case a gene called TUSC2, in order to replace that gene, and that we can show leads to very positive results in treatment of cancer patients. We're utilizing synergies that we've been able to demonstrate in vitro and in vivo in mouse systems, which allow us to essentially add on to established therapies. At the moment, we're focused on lung cancer, but we recognize that with tumor suppressor genes, there are multiple cancers that we can begin to address. Basic idea here is to take the tumor suppressor gene, in this case, the TUSC2 gene, encode that in a plasmid, package the plasmid in a lipid nanoparticle, which is then given intravenously and taken up by the cancer cells. There are mechanisms to be able to target the cancer cells. One is that lipid nanoparticles have a positive charge, and the second, and the cancer cells have a slight negative charge, while normal cells have really no charge. And the second is that cancer cells have an increased rate of pinocytosis and take up molecules like lipid nanoparticles at a much higher rate than normal cells do. Once inside the cancer cell, the plasmid will express the TUSC2 gene, leading to the TUSC2 protein expression, leading to death of cancer cells, but certainly marked decrease in proliferation of cancer cells. Turns out that the TUSC2 containing lipid nanoparticle, which we're calling Recorsa, has multiple activities. TUSC2 inhibits cancer cell signaling by inhibiting tyrosine kinases. It stimulates apoptosis, and it modulates the immune response. In our monotherapy trial, previously done with Recorsa, we've been able to show that Recorsa can lead to marked increases in TUSC2 expression. And here we have baseline and 24-hour post-treatment biopsies from 2 patients. Therapy trial looked at various doses of Recorsa. Stable disease was seen in 5 patients with some responses in 3 of them, and with showing decreases in the size of certain of the metastases. The interesting thing is the drug is very well tolerated in patients. There isn't an infusion syndrome, which is relatively easily ameliorated, but that appears to be the major side effect. Well, shown here is of a patient who, prior to starting Vicorsa, had 4 lesions in the liver and peripancreatic area, and those lesions seen on PET CT scan. They disappeared after 2 cycles of Vicorsa. The patient had a total of 12 cycles. Each cycle is 21 days, and had a total of 12 cycles before having disease progression. And actually had stable disease by CAT scan throughout. The second trial that was performed was taking advantage of the tyrosine kinase inhibition of Vicorsa. It was combined with Tarceva, which is an inhibitor of the EGF receptor, tyrosine kinase. And these are data from 7 out of the 10 patients in the phase 2 trial at maximum tolerated, what was believed to be the maximum tolerated dose. And you can see that there's actually been a number of responses in those patients who all were progressing coming into the trial, who had at least stable disease in the trial. One had a complete response. Several others had regression of various lesions. All these patients were heavily pretreated with number of prior, and they were consistent patients both who are EGF receptor positive in terms of mutations, as well as negative for EGF receptor mutations. So there were responses in both groups of patients. Importantly, there were several patients who had heavily were pretreated with Tarceva, and nevertheless had some level of response to the combination of Vicorsa and Tarceva, again suggesting that that this drug combination could be used in patients who are progressing on Tarceva. Tarceva has largely been replaced by a different EGF receptor inhibitor, Tagrisso. And here are preclinical data using the combination of Vicorsa and Tagrisso in a resistant cell line in xenografts in mice. And in the green and red lines, one can see that the addition of Vicorsa to Tagrisso led to increased activity and significantly increased activity in controlling the tumor in these mice. That's led to our CLAIM-1 clinical trial, in which we're evaluating patients with non-small cell lung cancer who have EGF receptor mutations and who have progressed on Tagrisso. Phase 1 trial, of course, they'll be evaluated at high. 2, they'll be treated, randomized between combination and Tagrisso, and continue Tagrisso monotherapy alone. For survival, we think this will be an important trial. We've also been able to combine Vicorsa with agents, both in vitro and in vivo in mouse systems. In this particular case, we've utilized a humanized mouse model that uses a human immune system. And you can see in this mouse model that there's an effect of Vicorsa alone. There's some effect from pembrolizumab alone, but there's a much dramatic effect in synergy with the combination of Vicorsa and pembrolizumab. And I believe this is due to effects on the immune system. We can show that Vicorsa increases the number of cells, increases the number of natural killer cells, and decreases the number of myeloid-derived suppressor cells that are found in the tumors. So we think those immune activities of Vicorsa are just what's needed to be combined synergistically with a drug like Keytruda. And that's led to further studies where we wanted to look at the combination of Vicorsa, Keytruda, and chemotherapy. And essentially that's the red line here in a mouse xenograft showing that addition of Vicorsa is dramatically beneficial. to the Keytruda chemotherapy combination. And our studies also showed that we still see the same immune effects when Recorsa is added, that we see the same immune effects as seen without the chemotherapy. This suggests that we can move to the first-line standard of care in future trials. In the present trial, this is led to the ACCLAIM-2 trial, and in non-small cell lung cancer patients whose disease has progressed after treatment with Keytruda. And then we have a phase 1 portion to find the maximum tolerated dose. But then in phase 2, we're randomizing patients 2-to-1 to Recorsa and Keytruda versus a more standard treatment for relapsed patients, docetaxel with or without ramucirumab. These patients will also be followed for time to progression. Also recognize that small cell lung cancer, in addition to non-small cell lung cancer, is an excellent target for Recorsa treatment, and we'll be adding that soon enough to our clinical portfolio. I want to spend just a couple minutes talking about our diabetes program as well, because it's a very promising program, although in preclinical stage. The basic problem, of course, is that patients with diabetes have to continue to take insulin, and the idea here is to be able to convert alpha cells in the pancreas into beta cells that do secrete insulin, and have that replace exogenous insulin treatment. There are a couple of novel aspects of this program. We're using AAV vector with 2 specific transcription factors that lead to the transition from alpha cells to beta cells in the pancreas. And we're infusing those cells directly into the pancreatic duct, so there's not systemic exposure to the AAV virus. And as a result, we can use relatively low titers of AAV virus and be effective. And the second aspect is that this is an autologous program. The beta cells produced are of the same, or obviously from the same patient. There's no allogeneic aspects to this, and thus there's no issues with rejection at all. This has now been moved into studies in monkeys to be able to then pave the way for clinical trials. This shows our key milestones through the first quarter of 2022 and then beyond to the rest of the year. We'll have a very busy time and lots of activity in all 3 of these areas. So a summary of our approach: we do believe that we're working with 21st century gene therapies and we're evaluating large markets and significant unmet medical needs. We're focused in the oncology trials and combination trials with top-selling drugs so that we can add to their benefits. We've certainly gotten recognition for this with 2 FDA fast-track designations, and we're continuing to explore new indications and partnerships, new indications in terms of new diseases like small cell lung cancer, as well as the potential for additional tumor suppressor genes to be utilized either in lung cancer or in other cancers as well. And of course, we'll be moving along our diabetes program towards clinical trials. Thank you very much.